When you think of what makes up a forest, most people think of the trees and other plants. However, due to rising global temperatures that are worsening severe storms and coastal flooding events, coastal forests as we know them are changing. To address how coastal forest ecology will change in response to increased flooding, scientists at the Smithsonian Environmental Research Center have developed the Terrestrial Ecosystem Manipulation to Probe the Effects of Storm Treatments, or TEMPEST experiment. These tanks will flood these plots with tens of thousands of gallons of water. You might be picturing a flood of mythological proportions, but it's more of a series of hose leaks. Over a day, these plots will be saturated with freshwater and saltwater to simulate freshwater and brackish water flooding. As a member of the Molecular Ecology Lab under Melissa McCormick, after the third annual flooding of the TEMPEST experimental plots, I collected plant and soil fungi diversity and abundance data. This data is used to better understand the response of mycorrhizae to flooding. Mycorrhizae are mutualisms between fungi and plant roots where each partner benefits from exchanging resources with the other. The vast majority of vascular land plants have this mutualism with fungi. I've drawn the plant cells here in red. There are many different types of mycorrhizal interactions. Arbuscular mycorrhiza, which is the most common type by far, ectomycorrhiza, ericoid mycorrhiza, and orchid mycorrhiza. All of these interactions entail different types of resource exchanges between partners and vary in their dependence on and specificity of their partner for establishment and survival. Different types of mycorrhizae are expected to differentially respond to flooding stress and saltwater intrusion. Our lab measured the percent coverage of all the plants in the TEMPEST plots. No leaf, flower, or blade of grass was spared. We also took soil cores from random areas of the plots, as well as near certain plants we were monitoring to identify the fungi later in the lab. The soils were taken back to the lab to be prepared for metabarcode sequencing, through which the diversity of fungi in the sample can be identified. With this data, we can better understand the varying impacts of flooding on different mutualisms and relate the response to flooding of one mycorrhizal partner to the other. This project is one of many efforts to help assess the trajectory of plant community composition in our beloved forests on the coasts.